Good morning and namaskar to all. I am Dr. Tikam Chundakal, presently working as an assistant professor in MLS University, Udaipur, Rajasthan. I am presenting a solution for COVID-19 treatment through nebulizer and inhaler-based ADTA chelation therapy. Coronavirus is a highly contagious virus that causes infection to human respiratory system by binding to lung epithelial cells. Coronavirus have spike proteins on their surface through which they attach to the host cells and gain entry into them. The infectivity of 2019 novel coronavirus is unimaginably higher than all previous known coronaviruses, and the reasons are largely unknown till today. I compared the protein sequence of spike protein from previous SARS coronavirus 1 of year 2002 and the recent 2019 novel coronavirus. And I found that the spike proteins of novel coronavirus have an important motif, called as RGD motif, in their receptor binding domain. And this RGD motif is not present in any other previously known SARS coronaviruses. This RGD motif was originally found in fibronectin and is well known for its role cell attachment and cell adhesion using divalent ions such as calcium and magnesium. A number of human proteins show interaction with these RGD motifs. One such group of proteins are integrins that show high affinity with these RGD motifs. I checked the expression of integrins in lung cells and found that expression of some integrins such as alpha V beta 6 and alpha 5 beta 1 and some others is very high. This indicated and suggested that integrins may have role in virus attachment with host cells. I did some bioinformatics-based molecular docking and found that 2019 novel coronavirus binds to the host cells integrins precisely through these RGD motifs. And a number of calcium binding sites have been predicted around and in vicinity to RGD integrins docking site. This implicated that calcium and other divalent ions may be playing important role in establishing these attachments. So, if we can somehow remove these bivalent ions from lung cells, we can prevent binding of virus with the host cells dramatically. And this can be done using ADTA, which is a well-known chelating agent. There is a therapy, called as ADTA chelation therapy, which has been previously used for the removal of toxic metals from body for the treatment of coronary heart diseases and neurodegenerative diseases. I propose nebulizer and inhaler-based ADTA chelation therapy for the treatment of COVID-19. Wherein ADTA will be allowed to pass into the lungs using nebulizer or inhaler. And ADTA will chelate these divalent ions in the lung cells, making them unavailable for RGD-mediated virus attachment with the host cells. I believe, in the absence of divalent ions such as calcium and magnesium, spike proteins of virus would not be able to attach to the lung cells. And in this way we can prevent infection of 2019 novel coronavirus and can attain a state of protection against these viruses. Apart from this, use of ADTA has additional benefits because calcium ions also constitute major part of viral envelope protein, and use of ADTA is expected to disintegrate virus. The most characteristic pathophysiological outcome of COVID-19 is excessive secretion of cytokines, also known as cytokine storm, in lungs, which lead to inflammation, fibrosis etc., and ADTA is expected to inhibit cytokine production. Antioxidant, anti-inflammatory and immune suppressive role of ADTA is also well documented in literature. Moreover, this therapy could be effective against SARS coronavirus 1, 2019 novel coronavirus and any other coronavirus that may emerge in future. The potential side effects of the therapy are hypocalcemia, proteinuria, and maybe some allergic reactions in some patients, but the benefits of this therapy outweigh its risks. Finally, I believe the nebulizer and inhaler-based ADTA chelation therapy present a novel, technically simple, quick, safe, affordable and efficient solution for COVID-19 treatment, especially in this global pandemic situation when any other effective treatment solution is inexistent. I am in process of developing and establishing a proof of concept and a working prototype model for this therapy, and for this, collaborations with advanced research labs, hospitals and startup companies are highly solicited. Thank you very much. Stay home, stay safe.